Welcome everyone to A-Level Biology. This presentation gives you an introduction to the layout of the course and the components that are required for success in this subject. So just to give you some of the flavour of our results, as you can see, we have achieved highly over the last 4 years. So basically our pass rate is 100%, that's above the national average. Our A to C percentage hovers around 90 to 95%, again well above the national average of 66, 67% approximately, and our A star to B percentage hovers between 60 and 70%, again well above the national average in those for that category. So you can see that we outperform the national average on all parameters. Lots of our students go on to very high achieving universities such as Oxbridge or the Russell Group. Last year we had lots of A stars, and we do regularly get 9 or 10 A stars from our cohort of about 60 to 70. We successfully send students to medicine and veterinary medicine as well, along with a whole raft of other destinations, including healthcare, but also including things like human sciences at Oxford and English literature at Oxford last year too. We are rated as an ALPS grade 2, which is outstanding on the ALPS scale. ALPS is a scale that essentially looks at how much value we add to our students. And we have been in ALPS grade 2, which places us in the top 10% of biology teaching in the country for the last 4 years. And we are consistently the highest performing subject area in the college. So as you can see, our results speak for ourselves. We are very passionate about achieving the potential that our students have. So the syllabus we cover, if anyone is interested to look at the syllabus in more detail, is the AQA A-Level Biology syllabus, which is available on the AQA website as the full specification, and it is A-Level 7402. We're going to give you a flavour of what's on the syllabus, but if you're really interested in knowing specifically what's in each unit, then you can download a copy for free from the AQA website. So there are 4 teachers currently responsible for A-Level Biology, and there's a lot of expertise in the department. So I myself, Nicola Manning, have won a National Teaching Award, a Pearson's National Teaching Award for my teaching of biology. We also have the Director of STEM, Tanya Hoffman, within our department, who heads up a lot of research, which we do within biology, when we're particularly focused on doing quite a lot of work on metacognition. And we also have Alison and Sinead, who are A-Level biology teachers. So if we look at the core content of the 2 years, as it is a 2-year A-Level course. So in the first year, we cover biological molecules, cells, organisms exchanging substances with their environment, and genetic information, variation and relationships between organisms. The first 2 topics we run up until Christmas, so they essentially occupy the autumn term. Topics 3 and 4 we run up until approximately Easter, where we have an exam and finish the year 1 content, and we move on to the year 2 content within the first year. So within the June and July of the first year, we've moved on to the year 2 content, and we also attend our annual field trip at Dawes Hall during that time. In year 2, we cover the units 5, 6, 7 and 8. So energy transfers in and between organisms, organisms responding to changes in their internal and external environments, which is essentially physiology, genetics, population, evolution and ecosystems, and then finally a unit that looks at the advances in DNA technology called the control of gene expression. We aim to finish the course by sort of Easter of the second year, so that we have a couple of weeks to revise before the exams, which are usually in June. So this gives you a flavour of the biological molecules unit. This is completely different from anything you've met at GCSE. It's new to everyone, and we look at a range of biological molecules in turn, and then discuss their importance, how they're adapted to their structure, etc. So we've got the carbohydrates, we've got the lipids and triglycerides, we've got the proteins, which include enzymes, and we've got a big topic of enzymes, a big chunk of material on enzymes to cover, because that fundamentally runs through the whole of biology. And then we've got DNA, RNA, ATP, and then we finish off by looking quickly at ions and water. Okay, so these molecules are covered in this first unit, and this is all new from GCSE. There's no prior knowledge on biochemistry. We start from scratch, and so it's really important, this unit, in underpinning quite a lot of the biochemistry that we do throughout the 2 years. The second unit that we start on in the first year is about cells. So you will start off by looking at the components of cells in terms of their organelles. This unit also looks at prokaryotic cells, bacterial cells, and how they differ from eukaryotic cells, plant and animal cells. The unit goes on to look at mitosis, so the way cells divide to produce new cells, and identifying stages within mitosis. There's then a big chunk of material on how material gets transported in and out of cells. And again, that's really important fundamental knowledge that weaves through the whole syllabus. So we do work on osmosis, we do work on active transport, facilitated diffusion, and something called co-transport, which is new from GCSE. And then finally, this unit ends up with a big chunk of immune system. And that's very current at the moment. So you look at virus structure. COVID-19 is essentially a retrovirus, the same as HIV. So we look at the replication, and then look at the immune system's defense against viruses and bacteria in terms of cell-mediated and antibody-mediated immunity. So there's quite a big chunk of material also on the immune system to cover in this first unit before Christmas. Christmas, we start on organisms, exchange, and their environment. So this is essentially a physiology unit. So you look at heart structure and the risk factors for coronary heart disease. You look at animal transport in terms of the structure of arteries, capillaries, and veins, and tissue fluid formation and tissue fluid return. You look at gas exchange, so surface area to volume ratios, gas exchange in fish, but also gas exchange in plants, gas exchange in terrestrial insects, and gas exchange in humans. This unit also includes digestion, which is essentially hydrolysis reactions, which overlaps quite a bit with the biochemistry that you've done in the first term. And then we do quite a large chunk on plant transport and how substances move in the xylem and the phloem. So also running in the second term of the first year is a unit on genetic information variation relationships between organisms. So essentially it starts off by looking at chromosome structure, DNA, differences between prokaryotic and eukaryotic cells, and then moves on to look at mutation, where there are mistakes in the DNA replication or copying mechanism. It then moves on to look at protein synthesis, something that students have met at GCSE, but they require a lot more information on. So we look separately at transcription and translation. We then move on to meiosis, because meiosis is one of the important things in generating genetic diversity. So unlike mitosis, we don't need to know all the phases. We just look at how it generates variation. We then look at the whole concept of genetic variation in more detail. Genetic variation is an important driver for natural selection, and we look at the types of selection. And a classic example here is the peppered moth, in terms of that's probably going to evolve into 2 different species of moth. And we talk about that process of speciation, which leads on from natural selection in the second year. We also look at how we measure genetic diversity in terms of looking at differences in amino acid sequencing in a given protein, looking at differences in DNA-based sequence in a given messenger RNA sequence from a gene, and then looking at differences in DNA-based sequence in genes. So there's a whole There's a whole section of material looking at how we assess genetic diversity. We look at bacteria and the effect of genetic diversity in bacteria, which is essentially one of the things that has led to antibiotic resistance, which is an example of directional selection, so a type of natural selection. And we have to do a practical about growing bacteria and looking at the effectiveness of various substances or various antibiotics against bacterial growth. And then this unit finishes with some material on species diversity index. So that's a way of assessing diversity within populations. There's some calculations, and then that looks at how modern techniques such as farming affect species diversity. And we start to talk quickly about the idea of conserving habitats to conserve genetic diversity. So that then ends the first year, and there will be exams on the first year before students are then allowed to progress into the second year of the course. So the second year starts with a large unit on energy transfers. So we look at energy transfers in a sense because some of that is linked to the work on the field course at Dawes Hall. But we look at the fundamental processes of respiration and photosynthesis, which are all about energy transfers. So they're two really big chunks of this unit. We also look at energy transfers within populations and how that links to modern farming techniques in reducing energy loss from food chains. And within this unit is also the nitrogen cycle and the phosphorus cycle. So this has lots of really big chunks of biochemistry in. Photosynthesis and cellular respiration are essentially biochemical processes, and so we need the underpinning knowledge from Unit 1 in order to successfully tackle this unit. Organisms responding to changes to internal and external environments is a unit that we look at after Christmas in the second year. So this is essentially physiology. So we start off by looking at responses to the environment involving receptors, so simple receptors, stimulus receptor coordinated effector response sequences. We look at synapses and nerves, so action potentials and resting potentials, how nerves transmit nervous information. We look at synapses and how drugs can affect synapses. We also then move on to muscle action, so how muscles contract and the physiological processes associated with that. This unit then looks at homeostasis, so the idea of keeping the body in balance in terms of blood glucose level, which links to diabetes, and we explore the different types of diabetes and the symptoms and the treatment for those different types of diabetes. In terms of homeostasis, we also have to look at osmoregulation, keeping the blood's water potential or water content within certain limits, and that's achieved by the kidneys. So we look at the kidneys in terms of excretion, removal of the waste product of urea, but also in terms of osmoregulation and regulating the water content of the body. So this is a really big unit and covers sort of the physiology that you would expect then to take further if you looked at a biology degree at university. This unit, Genetics, Population, Evolutions and Ecosystems, we run in the term before Christmas in the second year. Quite a lot of it is done on the field course, so you can see examples of surveying being undertaken in a meadow on the field course. So there's a lot of surveying of the river, the stream, looking at processes like succession, so primary succession and secondary succession, looking at predator-prey relationships. Those those things come into the first part of this unit. So essentially, quite a look at ecology, and moving on to look at things like conservation. We also look at genetics in this unit, so simple monohybrid crosses, looking at one characteristic, but we also then move on to dihybrid crosses where we're looking at two characteristics, and then further we look at things like autosomal linkage and epistasis, and there's some statistics comes in here, as well as on the field course. There's quite a lot of statistical testing that comes into the field course. We also look at genes within populations, which is what the formulae is. That's essentially something called the Hardy-Weinberg formulae, which allows you to make predictions about how gene frequencies, allele frequencies will change in the population. And then finally, we have a revisit of selection. So we revisit the types of selection that you met in Unit 4, and then we move that onto the process of speciation. So this unit runs up until Christmas in the second year. And the final unit, which runs after Christmas in the second year, is about the control of gene expression. So this is about all the advances in DNA technology that we've seen. So we start off by looking at mutation, how changes to DNA-based sequence can lead to genetic disease, and that links in with material that is done in Unit 4. We also look at the ideas of totipotency, pluripotency, and multipotency, and give you definitions for those words, and the idea of different types of cancer, benign and malignant, and how that links to genetic control in terms of tumor suppressor genes and proto-oncogenes. There's a big section then on DNA technology, so recombinant DNA technology in genetic engineering. We also look at advances in things like the Human Genome Project, genetic fingerprinting, and genetic counselling. Also, there's a section that looks at gene therapy, so how genes have been used to now treat genetic diseases, so using DNA as a treatment rather than conventional medicines. So this unit then ends around Easter of the second year, where you then move on to the revision phase, and this then essentially ends the content for the biology course. So in terms of assessment, we have internal assessment all the way through the course. So in the first year, we have end of topic tests every 2 weeks. So for example, we might teach carbohydrates and then we have a test, which is about 30 minutes, and they will be exam questions, and then proteins and lipids, and then we'll have a test, etc. So more or less every 2 weeks, students are having to learn the information. They're then tested using exam conditions. And what we've brought in with our work on metacognition is the idea that they're reflecting on their performance, and they're looking to become independent learners in the sense that they set their own targets for what they've got to learn, for how they've got to learn that, and how they're going to go about changing or enhancing their performance. We have a Christmas test, so a big test before Christmas, which tests the whole of the first term's content, which is essentially to check that people are on the right course, they're doing the right, they're making progress. And again, that's exam techniques and exam questions marked by the teacher, and feedback given individually to the students. At the end of the first year, we have full mocks. They are an hour and a half full-scale exams. We have a mock paper 1, which is essentially on the first year's content, and includes critical analysis and extended answers. And then we have a mock paper 2, which also includes critical analysis, but has a comprehension, which is part of the assessment as well. As we move into the second year, we have end of topic tests again. They tend to be every 3 weeks because the topics are larger. And again, we feedback to students and we get them to reflect on their performance. There will be a mock paper 1. So paper 1 is on the year 1 material, and that will occur sometime in the autumn term. And then there will be mock papers for paper 2 and paper 3 sometime in the spring term. Paper 3 also includes the essay, which we do a lot of work on in the spring term to prepare students. So in terms of the external assessments, this is what students will face at the end of the year. So paper 1 is on topics 1 to 4, essentially the first year's teaching. It includes practical skills, as do all papers. There are short and longer answer questions and an extended response question. It's usually 3 5-mark extended responses, and this counts 35% of the whole A-level grade. Paper 2 is again the topics from the second year, so topics 5 to 8. It includes practical skills. It includes short and longer answer questions, and there is also a comprehension on this paper. Paper 3 tests the synoptic component of the course, so tests understanding from years 1 and 2, all units. It is essentially structured questions, so we get critical analysis, although that does appear on all papers. We get lots of experimental and data response questions, practical questions, and then there is an essay to write for 25 marks, which is essentially one of 2 titles. So this is what we're preparing for throughout the course. So we tend to do the critical analysis, the comprehension throughout the 2 years, and we focus on the exam, the essay, at the end, because it is synoptic, so from the at the end of the course in the second year, from in the spring term, we will start to focus on essay writing skills. From the last slide, hopefully you've realised biology is not just recall of information. It's essentially very skill-based. So there is some recall of information, but there's also critical analysis, extended writing, math skills, practical skills. So there's a lot of other skills involved in biology. Now, there is a mathematical demand on the course. So we practice the maths throughout the course. 10% of the overall assessment, so 10% of the marks from all three papers, will be on maths. So again, as I said, we tend to practice those throughout the years. And at least 15% is about skills and understanding in relation to practicals. So when we do the practicals, it's not just to attain the practical endorsement, it's to gain these skills, which then can appear in all of the papers, but particularly paper 3 at A level. So this is essentially the weighting. We have 3 assessment criteria. Assessment AO1 is essentially about knowledge and understanding. So that's your learning of facts, and that's 35% weighting. AO2 is about context and quantitative and qualitative analysis. So it will be knowledge and understanding, but put into an unusual or unfamiliar context for students to interpret. And there will be, again, questions on practicals. There will be critical analysis of data. And then AO3 is all about evaluation, so making judgments based on practicals, making judgments based on material presented to the students, unfamiliar data. And so it's not just about learning the content, it's about practicing these skills as we go through, which essentially we do from day one in biology, so that we are preparing students for the fact that the exam is only 35% based on essential knowledge. The other percentage is about the skills that they should develop on the way through the biology course. So, alongside the biology A level runs the practical endorsement. So when you leave, you have an A level grade and a pass or fail at the practical endorsement. To be honest, the majority of students pass the practical endorsement, and they need to have covered 12 practicals over the 2 years. And they need to make sure that they've assessed certain skills, which are called the CPACs, and essentially they need to assess them and have attained them a number of times throughout the 12 practicals. But as we've seen, the practicals are important. They are examined within the A level. So developing the practical skills is an important component of not just the practical endorsement, but the A level. Okay, so these are the practical endorsements across the first year. So we look at an enzyme controlled reaction. We use microscopes to do a root squash, and we make drawings from those slides. You need to make a dilution series. That's an important technique in biology, and produce a calibration curve about water potential and osmosis. You need to investigate the permeability of membranes, and we use beetroot and anthocyanine for that. There is a requirement to do dissection, but it does not have to be an animal organ. So if people object, they can do a plant organ. So we tend to do hearts for the animal organ. But if you really don't want to do that, you can do a stem because that's an organ from a plant. And then finally, we finish with a bit of microbiology, which is essentially growing bacteria and looking at the effectiveness of antibiotics against bacterial growth. So this gives you a flavour of them. So you've got the enzyme practical, which involves hydrogen peroxide and catalase. You've got the root squash, and there is an image from a microscope showing onion cells in different stages of mitosis. You've got potato cylinders, and that practical is looking at osmosis, and that's where you have to make your dilution series. You've got beetroot permeability, so we've got a range of different beetroot cylinders. You can do this at different temperatures or try the permeability with different substances. We've got the heart dissection, but as I said, you can use an alternative of a plant stem, and you can see what we've got here is a plate of microbes, and we're using a mastering to look at the zones of inhibition and the effectiveness of antibiotics. Here we move on to 6 other practicals. So we use chromatography, a technique that students are usually familiar with, to isolate and look at the pigments from different plants. We look at dehydrogenase activity in chloroplasts using redox dyes, and we also look at dehydrogenase activity in respiration using redox dyes and single-celled organism of yeast. We also then use choice chambers, so we have usually wood lice, and we look at the effect of things like temperature, humidity, light level on the choices that wood lice make. Again, we have to produce a dilution series and use a colorimeter and a calibration curve to investigate diagnosis for diabetes, in the sense that diabetics have higher levels of glucose in their urine. These are not real urine samples, they're synthetic urines. And also on the field course in Dawes Hall, students are asked to undertake an individual investigation into a named environmental factor and the distribution of various species. So we look at things like distribution of certain plants within the meadow, linking to pH, linking to light nitrate levels, linking to light levels, things like oxide daisies, black knapweed, those sorts of things are common investigations. Also, you can look in the stream and you can look at various swimming mayfly larvae and other organisms that live, water beetles, etc. And essentially we look at their distribution in terms of current, in terms of oxygen level, pH, nitrate, phosphate level, and we do a lot of environmental investigation of environmental parameters on the field course. So as long as students complete all 12 investigations, and they essentially also complete the different skill levels, so there's various skills that they have to do within those practicals. For example, CPAC 1 is following a method, but they have to have successfully demonstrated that over the 2 years, then they will pass the practical endorsement. These are some of the practicals. So we've got chromatography of plant pigments. You can see the chromatogram produced, shows you different plant pigments. We've got a redox dye looking at photosynthesis, and we've got the idea that whether the redox dye decolorizes. Similar sort of thing for respiration. We've got the idea of decolorization of redox dyes. We've got the wood lice in their choice chamber, simple choice chamber looking at 2 conditions. And then we've also got one of the nymphs, one of the larvae that we find within the streams, and we can look and identify that, and then we can essentially look at its distribution in terms of oxygen level, current flow, pH, etc. Run through a number of frequently asked questions before I finish the presentation. So the best revision guides, we recommend the CGP guides. The CGP guides are usually the ones that students have encountered at GCSE, and they're very good for A-level biology. What we also suggest is that you make sure that you get the AQA guide, because essentially each exam board has their own CGP guide, and you just need to make sure that you're buying the biology AQA guide. And in fact, you've got the guide for the 2 years rather than just getting the guide for the 1 years. Okay, practical endorsement I think I've covered. So 12 compulsory practicals over 2 years essentially involves a number of skills within each of those practicals. And as long as you complete each practical and complete enough examples of each skill, you will pass the practical endorsement. In biology, each group tends to have 2 teachers. So I myself teach the whole of year 13, and then various people teach classes with me. And in year 12, Alison tends to teach the whole of year 12 with various teachers teaching with her. Now, the teachers have different topics, so we start on different units. So for example, in year 12, I will start on the biological molecules and Alison will start on the cells. So we run completely different units. We set different homeworks and different tasks, and we expect them to be completed for a week's time. So essentially, students need separate folders for each of the units, but then that will make up 8 folders over the course of the 2 years. There is maths content in the course. As we said, 10% of the overall assessment is maths, and we basically we do it throughout. So we always set exam questions for homework, and we often do a lot of it questions in the lessons, and essentially we make sure that there's some maths questions in there in every pack, so that we can then go through them with students, model our answers, so we're continually practicing the maths as we go through. Okay, so that ends the presentation on biology.